Hi, my name is Russ Huber. I'm a neuropathologist. I work for the Boston University CTE Center, and I'm also the director of the PTSD Brain Bank at the Jamaica Plain VA. My research interest is in examining tissue using a three-dimensional technique called clarity. The technique allows you to make tissue clear, and so you can see the different cells and different constituents that are inside the tissue in 3 dimensions. So the way that tissue clearing works is that you have to remove the lipids that are in the tissue. So what makes tissue opaque is the presence of lipids. And so in essence, to clarify the tissue, you're basically putting a detergent in that tissue, sort of washing the lipids out. And so the first thing you do is you stabilize the tissue so that it can handle that, and then you wash the lipids out. And then you have a material that is pretty clear at that point. And then after that, you label the different structures that you're interested in. We use different kinds of antibodies where we can look at different cells, things like astrocytes, neurons, all the different parts of the brain that we're interested in. We can also find the different types of pathology. For instance, we can see tau, we can see amyloid. These are things that are part of both CTE and also Alzheimer's disease. So we examine these underneath the microscope and can build a 3-dimensional image of these markers of the different types of cells and pathology. We use really fancy microscopes for this. One of the microscopes we have is called a light sheet microscope, and it basically just sweeps a sheet of light through the specimen over and over again to build a 3-dimensional image. And the other technique that we frequently use is called confocal microscopy. So both of these techniques that we use allow us to build large 3-dimensional maps of different cells and different pathologies. And the objective of this kind of work is to examine how the pathology spreads, and then also look at the different markers in the tissue that would indicate what kind of pathological process is going on. For instance, there are some markers of inflammation, neuroinflammation, which is a big part of both CTE and of Alzheimer's disease. And so we're trying to see what cells are expressing those different markers, and then that will give us an idea of what kinds of treatments we might want to use for the disease. And again, we're also trying to find just the regions that are involved and how it spreads, which is very important because in CTE, the first lesions you find in the frontal cortex, and then they gradually spread back into the hippocampus, which is the memory center. However, if you're looking at Alzheimer's disease, it's exactly the opposite. It starts in the hippocampus and then gradually moves out into the cortex. So understanding how the pathology progresses, and then also being able to look at the leading edge of the pathology, will give us an idea of what cells are at risk and what kind of damage that you can expect from the pathology.